All right, so in this video, we'll be going over digestion. So there's some digestion that's happening along multiple organs within our gastrointestinal tract. And so we'll talk about what kinds, how much, and what to look out for. And then in our last video, which will be after this one, we'll finish with absorption of those nutrients. So digestion, what is actually happening to our food? So we'll digest it, which is the breaking apart of these large molecules into their smallest pieces and parts. We will then absorb those smaller molecules into our bloodstream to be delivered to the rest of our body. And then whatever doesn't get absorbed is going to be eliminated through our feces. So all of this is happening along our organ system that we refer to as our gastrointestinal tract. And again, we finished the last video briefly looking at this little chart here. And again, the goal is to break down each of these large macromolecules into their smallest single molecule parts. So they're monomers. So for carbohydrates, we're breaking them down into monosaccharides, so individual sugars like glucose. For lipids, we're breaking them apart into their either monoglycerides, which is glycerol with one fatty acid, or separating the other fatty acids. And then for proteins, we're trying to break them down into their individual amino acids. So getting them down to their smallest pieces, parts, to then increase the ease in which we can absorb them across our cellular membranes. So the gastrointestinal tract is those series of organs that essentially makes a long tube for our food to follow as it goes through different stages of digestion. And so each of the major organs along this tract are going to be separated by a circular muscle called a sphincter. So that sphincter can close up to prevent food from moving forward or backwards, and then open up when food needs to move. Right? So the first stop along our GI tract is our mouth, right, or oral cavity. So you actually are beginning the process of digestion by chewing your food. Your saliva has a little bit of an enzyme called amylase, which helps with digesting those carbohydrates. And your saliva also is going to help soften your food to ease digestion later and ease it the way it moves down your esophagus. So the pharynx is also technically a part of this tract. This is this pink space you see in the diagram behind your mouth. But we're mostly just looking at it being transferred. So most of our digestion is happening in the mouth here. There's no real impact of the pharynx on your digestion that's happening in the mouth here. The esophagus is then essentially just a long tube for transport. There's no digestion or absorption happening along here. It's just to help get our food from our mouth down to the stomach. Then from the stomach, our food will move into the small intestine. From the small intestine, whatever is left will make it into the large intestine, also known as the colon. And then if anything makes it through the large intestine and does not get absorbed, that is what we eliminate through our feces. We also have various accessory organs as a part of this GI tract. So technically your teeth and tongue are considered accessory organs. The salivary glands are what produce and then secrete our saliva into our mouths. The liver and gallbladder are going to work together to produce bile that will be secreted into our small intestine. And then the pancreas also secretes into the small intestine, and the pancreas produces a majority of the enzymes. So a majority of the chemical digestion that is going to occur throughout our GI tract comes from those enzyme molecules, those proteins that the pancreas is producing and releasing into the small intestine. And so we'll take a look at the role that all of these play in more detail. And so along with the actual digestion part itself, when the food has reached a specific organ that is considered the gastric phase, we have our preparation phase, where our body is essentially preparing for the act of digestion. So thinking about food, our sensations that are related to food, so smell, taste, and touch, will stimulate our brains to start releasing signals for our organs like our stomach and small intestine to start preparing for food to be added. So these are anticipatory reactions or preparations. So those sensations that I was just talking about, smell, sight, touch, taste, produce signals that can stimulate our memory, which will also help reinforce those signals that are recognized by the medulla oblongata. So it's another part of our brainstem here. The medulla oblongata, in conjunction with the hypothalamus, which is really the like control center of the brain here, will then react to those sensations to send signals along our nervous fibers that will stimulate the secretions of things like hydrochloric acid into our stomach, basically telling the stomach food is coming, or you should expect food to come, and thus start preparing the internal environment for digestion. So it will stimulate the release of those gastric enzymes, right, and hydrochloric acid. So throughout this chapter, we're going to be talking about two different types of digestion that can be occurring along this GI tract. We have mechanical digestion and chemical digestion. And so whenever we're talking about mechanical digestion, this is more about physical force or the interruption of bonds, as opposed to chemical digestion, which is going to require the chemical input of energy, the chemical breaking apart of these molecules, the actual full breaking of bonds instead of just disrupting them. And that can be a bit confusing, but I'm going to try to point out specifically what is considered mechanical and chemical digestion along each of these parts. But just in general, keep these ideas in mind. Mechanical digestion is that physical breaking apart of food. So with things like force of chewing, or interrupting bonds, such as denaturing proteins, which we'll talk about in a little bit here. And then chemical digestion is anything using enzymes to actually break apart these molecules with a chemical reaction. So the first stop for digestion is going to be the mouth. There is digestion occurring in our mouth through chewing and saliva, as I mentioned earlier. So the saliva is going to soften and lubricate our food to help it get down our esophagus and into our stomach. But there's also a small amount of amylase, the salivary amylase. This is an enzyme. Anytime you see a term for a molecule that has the suffix ase, that is almost always going to be an enzyme. So a protein that assists in making chemical reactions happen. And then the prefix can help indicate what type of molecule that enzyme is specified for. So the chemical digestion of carbohydrates is done through amylase, an enzyme that helps break apart these carbohydrate molecules. So we do have a little bit of that in our saliva. So the breaking apart of sugars, the dissolving of sugars is beginning in our mouths. We then move down that lubricated, softened grouping of food from our stomach into our esophagus, and then food will pass the esophageal sphincter into the stomach. So there is a sphincter at the top of the esophagus as well. And so if you ever have like struggled to swallow something, or you need to like actually tell your body like swallow this thing, right? It's usually because your esophagus, your sphincter at the top of the esophagus is still closed. You can't get that food or drink past it. And so it needs to relax in order to open so that things can then pass through into the esophagus. And then there's another one of those sphincters at the bottom that will open to allow food into the stomach, and then close again behind it to protect the esophagus from all of the really acidic juices that are in the stomach itself. So here I have a closer look at what a sphincter looks like. You can take some time to look at this figure from the text. And so once we get to the stomach, how does digestion work here? So our stomach is composed of 3 different directional layers of the smooth muscle. And smooth muscle is any muscle that you don't have cognitive control of. You can't tell your stomach to flex, right? You have abdominal muscles, which you can control, but your actual stomach muscles, these that you see in the diagram here, we cannot control ourselves. However, another piece of important thing here is the directionality of these muscles allows for like squeezing to occur in every direction, which is really helpful for mixing the highly acidic gastric juice and thus allowing the interruption of some of the bonds that are holding these molecules together. Right? So when food reaches the stomach, it's going to be manipulated by those muscles squeezing and relaxing. These muscles can also stretch in response to food entering the stomach, which is going to help with releasing hormones and stimulating secretions that assist with digestion. Right? And as they constrict, that will also release hormones that stimulate the feeling of hunger, right? Because your stomach is empty. So anyways, that movement of the muscles is going to really help with this mechanical digestion. So as the muscles squeeze in their different directions, that's going to help mix our food with the gastric juices that are released from the glands around the stomach, which specifically is hydrochloric acid. And that makes the environment inside the stomach really, really acidic. So we only briefly touched on acids and bases in this class, but hydrochloric acid or acids in general are anything that can freely release protons. Hydrochloric acid is a great example of a molecule that freely gives up this hydrogen, making the environment of our stomach extremely acidic. And so the mixture of our digesting foods and that gastric juice is referred to as chyme. So if you ever have thrown up, you are essentially throwing up chyme, right? It's not just your food, it's your food and the gastric juices from your stomach. And so this hydrochloric acid is going to help with denaturing proteins and activate some of the enzymes that get released into our stomach. And so this denaturing proteins part is something that you might easily confuse with chemical digestion. But really, we're just interrupting some of the magnetic relationships in the chemistry of a protein to kind of release them from their really wound up tight form that they end up getting pushed into, and start to straighten them out and lengthen them so that we have access to the phosphodiester bonds that actually bind amino acids together. And then we also have chemical digestion in our stomach of proteins and fats. So we had amylase beginning the chemical digestion of sugars in our mouths. Now in the stomach, we have the beginning of chemical digestion for proteins and fats from the enzymes that are being released, specifically pepsin. So hydrochloric acid helps turn pepsinogen into the active form of this enzyme pepsin, which will help with that, not just interruption and denaturing of proteins, but then the actual chemical breaking apart of the chemical bonds holding those amino acids together. So here's a bit of a better look at what I'm trying to talk about with denaturing proteins versus actually using enzymes to disrupt their bonds to each other. So a protein is made up of a series of multiple amino acids bonded to each other. And most proteins are in the hundreds to thousands of amino acids long range. And this chain gets folded through different stages. So we have our secondary, or the primary structure is simply that long chain of amino acids. They will start to associate with each other and form hydrogen bonds, which are essentially like magnetic bonds that come from having a positive charge near a negative charge on these molecules that cause them to kind of attract to each other. And so they'll start to form these spiral helix shapes or these kind of bending pleated sheets. Those will then be further manipulated to bend into a particular shape with each other that we call our tertiary structure. And a lot of proteins end here. They can be active and do their job as in this lone tertiary structure state. But there are also a lot of proteins that are actually multiple of these polypeptides put together. So the quaternary structure would be 4 different chains of amino acids that are interacting together to actually do the job of that protein. So the example we have here is hemoglobin, which is the protein in our red blood cells responsible for carrying oxygen and carbon dioxide. And this protein actually is made up of 4 different chains of amino acids. These are 4 different polypeptides that are bonded together, and this is required in order for that hemoglobin to do its job. So the mechanical digestion of proteins in the stomach is coming from hydrochloric acid interrupting the bonds that are creating these tertiary, quaternary, and secondary structures. So interrupting where you see these dots, any kind of like magnetic pull on each other, trying to get them into this linear form where the actual bonds holding amino acids together can be interrupted. And then the enzymes will do chemical digestion to actually split these amino acids apart. So other important things to know about the stomach is that that hydrochloric acid that makes up the gastric juices, as I've said, is highly acidic, which for most cells would be extremely dangerous, including the cells that line your stomach. And so they're actually being protected by another type of cell whose whole job is to secrete mucus. And that mucus creates a thin layer around our cells that line our stomach, and thus protect them from the hydrochloric acid that they are very near to. And then lastly, we do have a small amount of absorption. So this is like the final stage of getting our food into our body. After it's been broken down into its small pieces, we can start absorbing it into our cells and then into the bloodstream. And so the only things that are being absorbed in the stomach are some water and then any drugs like medicine that you're taking. Those are designed to have their coatings dissolve once they reach the stomach and thus can be absorbed by your stomach lining cells. But the absorption of everything else is going to happen in our small intestine. So here's some look at the anatomy of our stomach tissue. So we have multiple cell types making up our stomach tissue. We have our mucus cells, whose whole job is to secrete mucus as protection against our gastric juices. We have parietal cells, whose job is to release hydrochloric acid and intrinsic factor. We have chief cells, which release pepsinogen, and that pepsinogen will become pepsin, which is an active enzyme. And then we have our endocrine cells, which are there to release these regulatory hormones, right? Those long-range chemical messengers, which help with a positive feedback loop of when there's food in the stomach, these endocrine cells release gastrin. Gastrin travels through the bloodstream, gets received by other stomach cells, telling them to release more gastric secretions. So release more hydrochloric acid, intrinsic factor, and pepsinogen. Another thing to keep in mind here is that because our stomachs are constantly shrinking and expanding, right? So when there's food in our stomach, it'll stretch. When food is gone, it can shrink. Right? So the stomach tissue here can actually fold, which allows for that stretch. So a bit more detail on what molecules are actually present in our stomach and our gastric juices, starting with that hydrochloric acid. So this is what is mechanically digesting proteins, is disrupting those hydrogen bonds that are producing those secondary and tertiary structures in order to allow this really jumbled, mixed, like think about like a piece of string that's been tied into a really thick knot. The hydrochloric acid comes in and helps detangle that knot until we get a long piece of string. And that allows for the enzymes to actually reach the bonds holding together individual amino acids in order to break them down. So this hydrochloric acid is also going to assist with activating that enzyme pepsin, so changing it from pepsinogen into pepsin, and keeping that low pH, which is going to help kill any invaders, any microorganisms that have come in with our food that we don't want to make it into our body. So pepsin is an enzyme that helps chemically digest proteins. So it's going to start breaking apart the actual bonds holding amino acids together. Intrinsic factor is going to help bind with B12, so one of our very important vitamins that will promote its absorption in the small intestine. So this intrinsic factor is essentially what is actually going to bond with the intestinal cells and thus bring in the B12 molecule, the B12 vitamin. Without this, the B12 vitamin wouldn't have any receptor cells that recognize it and could bind with it and thus absorb it. So the intrinsic factor is important as that like kind of flag, that recognition molecule, like, hey, I've got something important here, let us in. And then gastrin, which is a hormone that is going to, again, be part of that positive feedback loop. So food gets to the stomach stretches, that causes the release of gastrin. Gastrin ends up binding with other stomach cells, causing them to release more hydrochloric acid, pepsinogen, and intrinsic factor. That gastrin can also communicate with our brains to let us know that we're hungry. And then obviously there's the mucus there to just protect ourselves. So the neural control of digestion. So when there is food in our stomach, the signal that is going to be communicated to our brains through our nervous system, so through our nerves, is that the muscles are stretching. So any kind of muscle interaction is going to produce a nervous signal. So as the stomach muscles stretch, a nervous signal gets sent to the brain. The medulla oblongata translates that for the hypothalamus. The hypothalamus produces a response that gets sent back to the body. Okay? And so the response is an increase in secretion of that hydrochloric acid and pepsinogen. There are also some receptor cells that can bind with the peptides, so the like a few or a couple amino acids together, considered a peptide, so small proteins. And so when those peptides bond with the receptors on our stomach lining cells, that can also produce a response that is going to lead to more hydrochloric acid secretion. There's basically multiple systems that just kind of tell our bodies like food is here, we need to do work. And so it is a positive feedback loop, and that food is present, these things are responding, thus they tell the stomach to release more of the chemicals that are needed to deal with that food. So a quick question to consider here at the end as we finish up talking about the stomach. Why is the stomach separated on both ends by a sphincter? So remember, a sphincter are those circular muscles at the ends of each of our organs along the tract here. So why would that be important? Why would it be important that we have a sphincter muscle on the ends of our stomach? So I'll take a minute for you guys to think about this. Go ahead and pause. Okay, so hopefully you chose all of the above here, right? These are all situations that we would want to avoid. So gastric juices leaking into and irritating the esophagus would be bad. Remember, the gastric juice is extremely acidic. That would be really dangerous for any of our cells. That's why your stomach cells secrete mucus to protect them. And so if any of that juice was getting up into your esophagus when it's not supposed to, that's kind of when you feel that burn, or that like baby burp it's been referred to, right? Is when some of that splashes up that wasn't supposed to get up. The sphincters also help prevent food from leaving the stomach before that mechanical and chemical digestion has happened. So we need that to occur before it can get into the small intestine and finish its digestion with the enzymes present there. And then C, to prevent gastric juices from leaking into and irritating the small intestine. So same idea as with the esophagus, that hydrochloric acid could be dangerous if it's actually reaching the intestinal cells before the gallbladder has released bile to help buffer that acid. So all of these are important and help give us a reason to why these sphincters are here, right? Help explain why our systems have developed these closing and opening valves, essentially between each of our organs. Okay. So now moving into the small intestine. Okay. So once food has left the stomach and gets into the small intestine, there are a few things that are going to occur. So we're still going to have a bit of mechanical digestion, and we're going to have a lot of chemical digestion. But also what needs to happen is a very quick adjustment of pH. So that really acidic chyme coming out of our stomach needs to be buffered and become more basic as soon as possible, so that we can reduce the risk of damaging the intestinal cells that line the small intestine here. And so most of the solutions that are going to be put into our small intestine to help with digestion are going to be coming from the gallbladder and the pancreas. And I guess technically the liver secretes bile into the gallbladder, and then the gallbladder secretes bile into the small intestine. So the gallbladder ends up being kind of like a storage unit for bile. The pancreas is going to be releasing a majority of the enzymes that finish the chemical digestion for any food that we're going to absorb. So first off, that pH adjustment. So taking this really acidic chyme from our stomach, we want to buffer that and make it more basic as soon as possible. And so our liver produces these bicarbonate ions that becomes bile. So those bicarbonate ions help reduce the acidity from the acidic chyme. They essentially accept all those free protons and thus make it more basic. Okay. That bile is also going to assist in mechanical digestion, which emulsifies our fat. So in the same way that we had hydrochloric acid interrupting the bonds of proteins, emulsification is essentially interrupting the bonds of fats. So taking these larger complex triglycerides and starting to interrupt the way the fatty acid tails are connecting with the glycerol heads, so that we can get some separate fatty acid tails and get those glycerol heads only attached to a single fatty acid, becoming a monoglyceride. And then a large amount of chemical digestion is occurring. So the pancreas is secreting enzymes for all of our major molecules that we'll be ingesting. So we have our macronutrients being digested by proteases for proteins, lipases for lipids, and amylases for carbohydrates. Nucleases are enzymes that are there to break down basically DNA and RNA, all of the genetic material that is going to come, that does exist in our food, right? You're eating cells of other organisms, so there's also DNA. We don't talk about that as a nutrient, but there is an enzyme that helps break apart those molecules as well. So again, proteases are enzymes that break apart proteins. Lipases are enzymes that break apart lipids, fats. And then amylase is a protein that breaks apart carbohydrates. These are the important ones to pay attention to here. Okay. So this is a more detailed look than we had before at lipids. So again, most of the fats that we're getting in our diet are triglycerides, which would look like this. So we have our glycerol head with 3 fatty acid tails attached to it. Okay. And so the goal through our bile and that bicarbonate ions is to interrupt the bonds between the glycerol heads and these fatty acids so that they separate from each other and thus can be absorbed more easily. So, and this is called emulsification. So once the fats have been emulsified, they will then be forced together into what is referred to as a micelle. This is just naturally, they're going to start to clump up with each other as they're being removed from the glycerol heads. And so they form these little, it almost looks like a vesicle that our cells use to help transport materials across the cell. But it's a single layer of lipids where we have the hydrophilic heads pointing out, essentially protecting all the hydrophobic tails that repel water. So this just naturally occurs because the tails essentially are resistant to being near water. If you think about magnets again, if you point the opposite ends of magnets at each other, or I guess the same ends, so if you point a negative and a negative at each other, they're going to have a force that repels. You can't push them all the way. It's very difficult to. They naturally want to push away. The same thing happens with the tails of these fatty acids. When they are surrounded by water, they naturally are pushing away the water, and so they end up in this formation. And then in this micelle formation, they can just diffuse into the intestinal cells. So we have more hormonal control of digestion occurring here in the small intestine. So when there is, when that chyme is leaving the stomach and entering the small intestine, that low pH can be picked up or signaled. I don't know why the word is escaping me right now, but sensed essentially. Our cells can sense that low pH, and that will increase the secretion of pancreatic bicarbonate to thus buffer that chyme and make it more basic. And so that is in response to another hormone called secretin. So when we sense that low pH coming from the chyme in the stomach, secretin gets released, which goes and tells the pancreas to release bicarbonates, which will help buffer that solution. Lipids and small peptides, similar to how some peptides were bonding with receptor cells or with receptor proteins in the stomach. We have a similar thing happening here where lipids and small peptides will start to bond with receptors along the cells of the small intestine. And this will increase the secretion of CCK, which is going to increase the release of pancreatic enzymes as well as that bile from the gallbladder. So we have bicarbonate ions coming from the pancreas in response to the low pH of chyme. And then we have enzymes and bile, which are all going to play a role in the physical and chemical digestion of that food, being released in response to the presence of lipids and small peptides. So here's a look at how each of these different secretions, or where each of these secretions fall on a pH scale. So a pH scale runs from 0 to 14, with water being the neutral at a pH of 7. So the more acidic a substance is, the lower the pH is, and the less acidic or more basic a substance is, the higher its pH is, up to 14. So our water is 7, saliva is just a bit more acidic than water. The gastric juice in our stomach is extremely acidic at a 2. And then the pancreatic juice in bile is just a bit more basic than water is. So we buffer this gastric juice until it is completely safe and is no longer going to damage the intestinal cells that are lining that small intestine. This is a really helpful table to use as a reference when trying to keep track of what is happening, what is being secreted, where along this gastrointestinal tract. So I highly encourage you to come back and study this table if you're getting confused with where things are happening, or even recreate this table yourself and then come and double check with this. That would be a really good study tool. Okay. So along with digestion, most of the absorption that happens throughout our GI tract actually happens in the small intestine. So the food that is actually going to end up in your body is being absorbed in the small intestine. So the intestinal cells that line the small intestine will start actually collecting molecules that make it into your bloodstream. Right. And thus, in order to perform this function, the cells that make up our intestinal lining or our intestinal tissue are very specialized for just that purpose. So the general idea here is we have lots of different folds occurring, and every time the tissue or cells are gaining more folds, they're essentially increasing the amount of surface area that that cell has, and thus increasing the amount of contact that the cell has with the food that's moving through our digestive system. Okay. So the tissue itself has these large folds. The smaller bits of tissue end up folding as well into these villi. And then each of the individual cells have folds that come from these extensions of the cell membrane using protein fibers called cilia, called microvilli. So we have these circular folds of tissue. We have these smaller tissue folds, which is the bumps you see here. This is just zoomed in on one of those. So we have these smaller tissue folds called villi. And then we have these little tiny folds that are a result of extending the plasma membrane with protein fibers called cilia. And so all of this just increases the chance that food is going to, or that the molecules from our food are going to interact with the cell membrane and thus either diffuse or get brought in with a protein into the intestinal cells, and then eventually into our circulatory system. So lastly here, just another helpful figure to keep in mind or to refer to when studying or trying to track where these different processes are happening, essentially showing where digestion and absorption is occurring along the process of our gastrointestinal tract. So every time you see these little white dots showing up, that's where digestion is occurring. And then the less color that you see on the line is showing more and more absorption. So for carbohydrates, we have a little bit of digestion happening in the mouth, not much, if any, happening in the stomach, and then a lot of digestion happening in the small intestine, as well as a majority of the absorption. So by the time that your food is leaving the small intestine, any carbohydrates that it was going to absorb have been absorbed. Fiber is a much more structurally dense version of a carbohydrate that we don't produce enzymes to break apart. And so that fiber is not being digested in any of these organs. There may be a little bit of digestion of fiber in the large intestine, depending on which bacteria are existing in your large intestine. And then we don't absorb very much of the fatty acids that come from that. Most of it is just being excreted. Protein, we don't have any digestion of protein in the mouth. We begin the digestion of protein in the stomach, and then much more of that digestion is occurring in the small intestine, and it is finally absorbed in the small intestine as well. For fats, we do have a tiny amount of salivary lipase, so the enzyme responsible for chemically digesting fat. There is a little bit of that in our saliva, but not much digestion would occur here. No digestion of fat is occurring in the stomach, and then majority of that digestion occurs in the small intestine, where they are also absorbed. Okay. So essentially all of the nutrients that we're going to get from our food and into our body, all of those will have been absorbed by the time they leave the small intestine. Everything else that's making it to the large intestine is essentially just feeding the bacteria in your microbiome and being softened and formed into the poop or feces, which is going to be excreted later. That's all the waste from the food that you did not get use out of. So yeah, that's all for this part of chapter 3. I'll see you in the next video focusing on the actual process of absorption, so getting these molecules into our bloodstream, into the cells that need them.